Today we will be looking at an article on the role of PFK2 and insulin in the regulation of glycolysis in cardiomyocytes. My name is Beth Scott.
And I'm Adele Bernard.
Heart muscles need to work at all times, no matter if we're eating an ice cream cone or sleeping for 8+ hours. In these scenarios of high energy versus low energy, our cells are managing metabolic flux.
So diabetes, patients have challenge managing metabolic flux, largely due to a hormone called insulin. So let's look at what things look like in a normal patient. We've got insulin in the bloodstream, shown in green, and it binds to insulin receptors in dark green. This helps the uptake of glucose into the cell, which is a precursor to glycolysis. And we see those receptors being activated in yellow. In type 1 diabetes, however, insulin is not produced by the body properly. And then in type 2, insulin receptors don't work properly, and glucose concentration remains high in the blood in both cases. That means that glycolytic activity is going to be lower, and these cells are going to need to gain energy through other energy sources like fatty acid breakdown, which can lead to cardiomyopathy. So this study is significant and important because cardiomyopathy is the leading cause of death in diabetic patients. The researchers in the study, in order to study this, basically created some type 1 and type 2 diabetic mice by using streptozotocin as a drug to do type 1 and a high-fat diet to simulate type 2 diabetes. And this is to help them understand glycolytic regulation. So in this paper, they specifically focused on PFK2 presence and expression because it is known to be a crucial regulator in cardiac glycolysis. Okay, but where does PFK2 come into play? So to give you a broad overview, when insulin and blood glucose levels are high, this is going to lead to protein phosphatase 1 to activate the PFK2 complex, which then leads to more fructose 2,6 bisphosphate, a positive glycolytic allosteric effector, which is going to further activate glycolysis. So we can see that since insulin is upstream of PFK2, if we have low or no insulin, PFK2 is likely to not be as present. All right, back to the study.
Control mice were compared to multiple experimental mice groups that simulated diabetic insulin levels using drugs, overnight fasting, and high-fat diets. Overall, the study found that PFK2 is less expressed in diabetic mice and degraded in the absence of insulin. One experiment that led to this conclusion was an experiment analyzing mice that were fed low-fat diets and high-fat diets, which the researchers claimed stimulated type 2 diabetes. And the amount of PFK2 that was present in the cardiac homogenates of the control, the low-fat diet, and the experimental mice fed the high-fat diet. The two groups were analyzed after 7 days and 21 weeks on their respective diets by extracting heart cells. The cardiac homogenates were analyzed using Western blot SDS Page with PFK2 antibodies. As we can see from the figure shown, levels of PFK2 were significantly reduced in the mice fed the high-fat diets than in the control mice. As a reminder, if PFK2 levels are lower in the high-fat diet mice, this means that glycolysis is less likely to proceed towards pyruvate. Something else must create the energy for the heart cells of the high-fat diet mice, such as lipids. If these high-fat diet mice are modeling type 2 diabetes, we see how type 2 diabetes may lead to heart disease. And just so you understand, the bar graph represents the PFK2 amount per 100 micrograms of total protein. And we see that in the high-fat diet, it is much lower. The study also found that PFK2 was degraded when there was no insulin signaling. The researchers cultured adult mice healthy heart cells in various conditions. C is the control group, where cells are exposed to normal, non-diabetic heart conditions. INS cell culture did not have any insulin. HG stands for the high glucose concentration culture, and HF is the high fat culture. These three experimental cell groups were used to determine under which conditions PFK2 would be degraded, if any. As you can see in the top of the figure, these groups were modeling type 1 diabetes where there is no insulin, high blood glucose, and a high fatty acid content. The group found that in the absence of insulin, PFK2 is quickly degraded. They did a Western blot STS page and calculated the cells without insulin had a 40% decrease in PFK2 protein content overnight. In Figure B, they used a phosphoantibody and saw a similar decrease in phosphorylation of a necessary kinase. Figure C shows that they proved using a Western blot that insulin is necessary for PFK2 presence. They confirmed this using an insulin-like growth factor, GF1, and saw the growth factor had similar results. In Figure D, we see that they use cyclohexamide and wortmannin. Cyclohexamide inhibits protein translation. Wortmannin inhibits an insulin-dependent pathway, P13K. They found that the cells with wortmannin had significant decrease in the half-life of PFK2, which you see in red, compared to the control in black with only cyclohexamide. They used these findings to come to the conclusion that PFK2 is degraded in the absence of insulin.
So to summarize other experiments from this study, we also saw decreased PFK2 expression with overnight fasting and drug-induced type 1 diabetes. Overnight fasting also increased glycolytic intermediates that were prior to PFK1, and PFK1, if you don't remember, is regulated by PFK2 in glycolysis. As we can see from this figure from our paper, those early glycolytic intermediates ahead of PFK1 are shown in bright red, which means that there's been a % increase in their presence compared to regular normal mice in glycolysis. And this is especially significant for diabetics because we can see that one of our intermediates is actually part of our glycogen pathway, and so if mice or diabetic humans need more energy through something like glycolysis, instead they're actually more likely to be storing energy for later, which does not help in the release of energy. So some overall takeaways were that low levels of insulin led to lower expression of PFK2, no matter if that came from a high-fat diet or overnight fasting. And as we explained at the beginning of the presentation, type 1 and type 2 diabetic heart cells will not be able to have normal insulin response. And so those low levels of insulin or low reception of insulin is being experienced in diabetic cells, which suggests that there's also lower expression of PFK. And secondly, that the lack of insulin in heart cells led to the degradation of PFK2. So this further suggests that without insulin, we don't have PFK2, further decreasing cardiac glycolytic activity. The significance of this research is that the study showed how PFK2 levels persistently stayed low regardless of the fed and fast state. And so whether they use mice with a high-fat diet or induced diabetes, we saw that PFK2 levels were low regardless of their fed conditions. And this is significant because of this metabolic flux that diabetic people and normal people need to adapt for. And this is integral in a healthy body because we need PFK2 levels to rise to undergo glycolysis when blood glucose levels are high. We can use glucose for energy to keep our heart muscles moving. So to wrap up, and decreased levels of PFK2 can lead to decreased ability to respond to metabolic flux, and this may increase risk of heart disease over time. So some next steps that we suggest are if researchers could find ways to modulate PFK2 levels independent of low levels of insulin due to diabetes, this could be extremely beneficial to prevent cardiomyopathy in diabetic populations. Let us know if you have any questions. Thanks.